So hello everyone. I think we only have 20 participants. We'll wait for a few more minutes, wait for your friends to join. Meanwhile, I'll share the screen. Okay, while waiting, I just wanted to ask y'all, any of y'all having problems with your Ubuntu still? Did y'all try out? I think Dr. Siva gave some links the last week, on Monday, for y'all to try out, look through your basics in Linux again. Did y'all manage to go through?
Yes, doctor.
Anyone having any problems, please let us know so that we can troubleshoot it before your project starts. Right? Right. I think I'll start with today's lecture. Okay, so today's class is going to be about similarity, homology, divergence, and convergence. So this has got to do a lot with evolution, and I'm very sure you've heard of this term and probably have studied a bit about it in genetics, but today's lecture is going to be sort of like a recap, and I will introduce new terms to you, which is relevant to comparative genomics. So as you can see this picture here, you see two people who look very similar. Only the only difference is the height, but you can see that the facial features are similar. And so this is actually similarity due to inheritance because they're actually related, they're siblings. And on the right, you can see two impersonators of Elvis Presley, right? And clearly they're not siblings or related, but they're similar, and superficially similar because they're impersonating a famous person. And this is due to other factors, okay, sort of like environmental factors. So this is what we're going to be looking into today, not about them, but in terms of genes and proteins that we are interested in. Okay. Right, so as I mentioned, this has got to do a lot with evolution. What is evolution? Is basically a process that results in heritable changes in a population, which is spread over many generations. And this process is actually directional, and essentially irreversible changes occur, and this gives rise to a more ordered or more efficient form of the organism. Okay, as you can see how humans have evolved, we have become more and more efficient, more and more organized, efficient, and superior compared to our ancestors. Okay, and this may occur as a result of natural selection, genetic drift, or both. So it can be because of stresses, environmental factors, or just by chance. Okay, so it could be either of these or both, but these are the minimum requirements for either for the process. So why are we very interested in studying evolution and evolutionary process? So this actually helps us understand the history of life, like how humans came about and why we have evolved over the years, and how this is actually happening. This is a very important question that biologists are interested in, and by studying evolution, we will be able to answer these questions. And it also lays the foundation for much of what we know right now in immunology, in genetics, in antibiotic resistance, human origins, and also how, over the years or over the centuries, how organisms have actually adapted to the environment, which is ever changing. Okay, so by studying evolution and similarity and homology in organisms, or similarity in genes and proteins, we can try to make sense of why it happened, and we can also, start predicting of what can be next or what's going to happen next. Right? So these are the important concepts in evolution that you need to have an idea about. Of course, your exams are not going to test you these things because, as I mentioned, your exams are very practical oriented. But to be able to carry out your project and to have the right questions to answer, you need to have an idea about what is similarity, what's homology, what is divergence, what is convergence, right? So these are the 4 concepts that we're going to look into today. And I've also given a link here. It's quite an interesting page. You can go through it later. They also explain same things, but more with a lot more illustration. So you can go and have a look at it later. Okay, so the first term, similarity or similar, okay, is also synonymous to the word analogous. Okay, and this actually means similarity in organisms from different lineages or from different ancestors. So these organisms don't share a common ancestor, but they still have similar superficial features. For instance, a bird which has wings and an insect which has wings. They don't come from a common ancestor, but they both have, if you look at the overall structure of the wing, superficially, meaning what you can see, right? Not the bones and all the other stuff, just superficially their wings, they look very similar and they have the same function. The wings help them fly, right? Okay, so this is an example of an analogous structures. Okay, they superficially look similar, they have similar function, but the organisms don't share a common ancestor. Okay? So this similarity is also known, and it is actually due to convergence or convergent evolution, which means you come from completely different lineages, but because of environmental factors, environmental factors or stress, they need to adapt to the environment, and they, you know, they get this ability through this structure that they're able to fly, right? So these structures are superficially similar, as I said, and they have common function, and these are known as analogous structures. So these are the terms that you should sort of understand. Similarity always refers to different lineage, right? It means organisms don't come from a common ancestor, and they have very similar superficial structure and a common function, and these are known as analogous structure. Right, moving on to the term convergence. Right, so this is actually the act of converging and especially moving towards union or uniformity. So now if you look at the definition of convergent evolution, this is actually a process in which 2 distinct lineages, means completely different ancestors, they evolve a similar characteristic or feature, or a limb, okay, independently of one another, meaning they don't rely on each other. Because of the stress and environmental changes and factors, they need this to survive, for instance, and so they actually get this structure or limb to be able to do it. In terms of you're looking at genes and proteins, they actually adapt to have new genes that can do certain functions. Okay, so analogous structures actually result from convergent evolution, and this often occurs because both lineages face similar environmental challenges and selective pressure. Okay? And so this is also known as a functional convergence, as they usually converge due to the need of a similar function. Okay. Right, so the next term, homology, is also, of course, if you look at dictionary, it means similar. Okay, so this is actually similarity inherited in organisms from common ancestors, no? The same evolutionary descent. So originally it was used to compare phenotypes, especially doing comparative anatomy or physiology. And these homologous structures may not perform the same function, but they have a common origin or structure internally. So this is an example. So this is a human's hand, cat, whale, and a bat. So you can see that evolutionarily they share a common ancestor. Okay, superficially, if you just look at the outer, human's hand look very different from cat's legs or whale's fins or bat's wings, right? But if you look at the internal structure, the anatomy, they actually have the same bones. We all have the same bones. We have humerus, radius, ulna, carpals, metacarpals, and phalanges. All of them have the same thing. It's just that they have a different morphology, or morphology looks different, but they have the same things as us. And superficially, if you just look at it from the outer view, they look very different, right? And usually, they don't have the same function. For instance, humans, we use our hands to do things, but we don't use it to walk or fly, right? Similarly with cats, they use their legs to walk, to run. Whales for others, and bats, they are using it to fly. So they may not perform the same function, but they have common origin or structure internally. Right? So these structures are known as homologous structure, and this is just the opposite of analogous structure. So, and this is actually a result from divergence from a common ancestor. So this is known also as divergent evolution. And another example given here is between the four limbs of penguin, alligator, bat, and a human. As you can see, we all have the same bone structure, not structure, the same bones are present, but they look morphologically different, the internal structure. Okay, superficially also they look very different, and we all have different functions for these limbs. Okay, so these terms are very important. You should know that homology also means anatomically similar structure, and we all have common ancestors, and it may not, however, may not have common function. We may use it for other different purposes, but these structures are known as homologous structure. Right? So homology, as you can see, I've given examples of organisms and limbs and wings, right? But this is not actually limited just to physical structures. So in comparative genomics, we are very interested in seeing similarity or homology between genes and proteins from similar species or similar genus, or from even different kingdoms, right? So this was then later adapted to the comparison of genes and proteins. So if you look at homologous genes, you're actually looking at genes that share a high or that share a similar sequence, okay, which means it has high sequence similarity. So an example is Hox genes, which is actually found in all multicellular animals. And this gene actually determines head to tail axis in embryonic development. And you can see this in all animals, multicellular animals. So if you do a alignment of these genes, you can actually identify what is the sequence similarity, how many percentage similarity there is between Hox gene from a human versus a Hox gene from, for instance, a rat or a monkey or rabbit, right? We can actually do this kind of analysis as well, right? Apart from homologous genes, we could also study amino acid sequences, which means we're looking at homologous proteins. So they also share extensive sequence similarity, structural similarity as well as chemical similarities. So usually when you see proteins with similar sequence, right, very high sequence similarity, you can be sure or almost sure that the structure of the protein is going to be similar, and then the chemical function of the protein is also going to be similar. Okay, especially if you notice that there is high sequence similarity in the domain, right? The domain is the functional component of a protein. So if you see like 90% similarity of sequence, amino acid sequence in the domain of, for instance, an enzyme in human and an enzyme in a rat, you can be sure that the structure of the enzyme will be almost the same, or at least the catalytic site will have almost same structure, and then the enzymatic activity will also be similar, right? So an example here, I've actually have an alignment over here, is cytochrome C. So this is actually found in almost all living cells, which is involved in cellular respiration. And I've shown a sequence alignment of human cytochrome C, which has been aligned with rat cytochrome C. As you can see, it has almost 90% similarity. You can see it's all the same. M, G, D, B, E, it's all the same. Only when there is a plus sign or there is a gap, then you know there's either a mismatch or a substitution or an insertion or a deletion. Okay, and if you this is the first sequence, and this has about 91% similarity in the proteins, and the second one is an alignment of human versus yeast cytochrome C, and you can see that the similarity is a bit lower. It's about 64%, and as you can see, there are a lot of gaps, mismatches as well. So this is some of it are just mismatches, some of it are gaps, and yeah. So this is an example of how in comparative genomics we would actually study similarity or homology between genes or proteins. Okay, so moving on, we also have specific terms for homologous genes. They are also known as homologs, and they can be further divided into two other forms, right? One is known as paralog and another known as ortholog. Okay, and this is believed to arise. Homologs are believed to arise from a common ancestral sequence. It means the genes have a common ancestor, and when the species diverges from a common ancestor, you have homologs. Okay, and so there are two types of homologs, paralog and ortholog. And we look at the difference between these. Okay, so how do you distinguish an ortholog from a paralog? Okay, so orthologs are actually homologs or homologous genes or proteins that come from two different species separated by a speciation event. Okay, so which means the genes or proteins still share a common ancestor, but during evolution it has actually split into two different species, right? So each species have a copy of the gene, and that gene is known as a homolog, or that protein is known as a homolog protein, right? So the key here is it's from two different species separated by speciation event. And if you look at paralogs actually are homologs which are present within the same species, and this is usually is due to gene duplication event. So this is also pretty common. If you even look at bacterial genomes, you will notice there are multiple copies of the same gene present in the same genome, right? So this is an example of a paralog. Right, I cannot think of any examples right now, but I can in the next class, I can give you some examples, but I think in my later slides have, right? So this, as you can see, I've also illustrated. So this is an ortholog where you have a common ancestor, they come from the same genus, and due to speciation event, they form two different species, and each species has the copy of the gene. So the gene is then known as a ortholog, right? And if you're looking at paralogs, so this is, for instance, this organism A and it has gene A, right? After a gene duplication event, the gene is actually, another copy of the gene is made, and in the same genome, or in the same organism or in the same genome, you have two copies of the same gene. So these genes are known as paralogs, right? Okay, so moving on to give you a bit more description about orthologs. In evolutionary terms, robust identification of orthologs is actually important to reconstruct the gene repertoire and also the gene order in ancestral genomes. So with respect to functional analysis, orthologs usually retain the same function because there's only one copy of the gene, right, in the genome. They normally maintain the function unless the function is no longer needed for the survival of the organism. If it's not needed, then eventually it will disappear from the genome, right? But if it's still required for their survival, it will maintain the gene over the years, over the decades, over the centuries, and it will still maintain the same function, right? So I've given this explanation here. So consider a gene or its product, let's say it's an enzyme, in an ancestral species that was responsible for carrying out some essential biological function, right? As long as the progeny of this ancestor carries a single copy of the gene and does not evolve or acquire an unrelated gene which is capable of doing the same function, the original gene, right, this will continue carrying out the same function. So it's either the gene no longer is needed for survival, it will be lost, or another gene is acquired from another source, and that gene is able to do the same function, then this original gene will either be lost or will evolve to do another function, right? So this actually puts the ortholog under a very strict evolutionary constraint and makes them perform the same function because there's only one copy of the gene and it requires it for survival, right? So this is orthologs. An example of orthology is insulin, right? The genes coding for insulin in humans and mouse are actually very similar and they're known as orthologs, okay? They have a common ancestor gene in the ancestor species of human and mouse. So moving on to paralogs. So in contrast, paralogs usually tend to evolve and adapt or get new functions. As you remember, paralogs are actually due to gene duplication events. So you will have multiple copies of the gene. This example just shows, 2 copies of the gene. Sometimes you have up to, 8, 10 copies of the gene. So in that case, that gene which is present, 10 copies of the gene, each gene doesn't need to function the same way in order for it to survive. As long as one gene still does the job, the other 9 genes can evolve to do some other function, right? So this is especially very important when you have environmental challenges or environmental factors which are stressing the organism. These paralogs can actually adapt and start having a new function to help the organism survive better in the new environment, right? So once paralogs emerge as a result of gene duplication, one copy of the gene is actually free to mutate and acquire a new function as there is no selective pressure. As long as among the, let's say it has 10 copies of the gene has been made, as long as one of it still is doing the original function, the others can evolve to have some other function. Okay, so we have noticed that in each sequence genome, between 25 to 80% of the genes actually belongs to families of paralogs. So as I said, most genomes have a lot of copies of certain genes. Okay, and so usually the genes evolve and do some other function which is beneficial for the organism, right? So classic examples include animal olfactory receptors or nuclear hormone receptors. All right, and this includes receptors for thyroid and steroid hormones, retinoids, and vitamin D, and vast families in which an astonishing repertoire of specificities evolved as a result of multiple duplication events. So the duplication event is not necessarily just once. It could happen over generations, multiple times, and you finally get like many copies of the same gene. And if that happens, these genes are known as paralogs, right? So an example of paralog is hemoglobin. So you all know that hemoglobin is a protein complex which helps with transport of oxygen in human body. And it's actually made of 4 protein subunits, which are your alpha, beta, gamma, and delta, right? And these subunits, right, are actually paralogs of each other, okay? And they have a common ancestor gene, right? Right, so now moving on to alignment. Now, as I mentioned, one of the best methods to look for homologs or to look for similar genes, similar proteins, is by doing alignment, right? So one of the main objectives of DNA and protein sequence analysis is to identify homologous sequences and then to employ whatever information you have on the sequence and structure to predict biochemical activities of the protein and biological functions of proteins and non-coding sequences. So this is what we do. We actually study model organisms, the genome of model organisms, annotate the genes, identify the function of the proteins, and then use that as a reference or a template, and we do genome alignments or sequence alignments with other genomes, new genomes which are sequenced, and then using whatever knowledge we have of the reference, we sort of transfer the same knowledge to the new genomes that are being sequenced. So if you see that, let's say your genome A is your reference from a model organism, and you just recently sequenced another species from the same genus. Okay, they are quite closely related. So all you need to do is a genome alignment with the reference, right? And if you have very high similarity of sequence, you can be sure that most likely the gene will function the same way, the protein will function the same way, and the protein will have a similar structure, right? So with that, you can you can actually do all these predictions of how or what the gene is going to do, or what does the protein do in the second organism that you have sequenced. Right, so how do you determine homology? You already know the answer, through sequence alignment. So alignment actually helps you estimate homology by sequence similarity, and it specifies which positions in 2 sequences match. So as I've shown here, this is a very short stretch of sequence, nucleotide sequence, and you can see that in the first, right, there are only 2 matches, and the rest are all mismatched, and the last one is not aligned, right? And then the second sequence, right, it's the second stretch, you have 5 matches, 2 mismatches, and 1 not aligned. Here you have 7 matches, 0 mismatches, and 1 which is not aligned, right? So the not aligned could be an insertion or can be a deletion, depending on which sequence you're using as a reference, right? So mutations such as insertions or deletions and substitutions can be observed during sequence alignment. So you can also look at SNPs, or single nucleotide polymorphism, when you're doing alignment. As shown here, the one in blue, this is a SNP, right? There's a change in one single nucleotide. And this, the red, is actually an indel. So it could be an insertion. In this stretch, it's known as insertion. In this stretch, it will be known as a deletion. Okay, it all depends with respect to the ancestor sequence. So you must have a reference genome or a template to refer or to align it against. Right. So Similarly, you can do the same thing with proteins. You can do sequence alignment with amino acid sequences. And in fact, homologs can be easier identified with alignment of protein sequences. So I believe you all should know of these terms, synonymous mutation and non-synonymous mutation. So synonymous means silent mutation. So even if there is a change in the nucleotide sequence, there is no change in the amino acid sequence, right? You remember your codon table? Only the last base, when only the last base changes, the amino acid actually doesn't change. So if you have codons where only your last nucleotide is actually changed, there won't be a change in the amino acid sequence, and hence the sequence similarity is the same, or you have high sequence similarity. But if you're looking at non-synonymous mutation, means there are changes in nucleotide sequence in either the first or the second base, which results in a change in amino acid. So we are actually interested in non-synonymous mutations because that, right, will change the amino acid sequence, and hence it will change also the structure of the protein or the domain. And a change in structure of protein will also mean change in function of the protein, or a modification in the function of the protein, right? So this is what is explained here, right? So a real problem actually arises when the similarity between two given sequences is much lower. So if you're looking at sequences where it's 90% similar and all that, you can be quite sure that the genes function the same way or the proteins function the same way. But if you have sequence similarity, which is like, say, 20%, 30%, how are you going to be sure that the genes or the proteins are going to function the same way, right? So here's an example. So let's say two protein sequences have 10% identical residues and an additional 8% similar amino acid residues. So a total of only 18% similarity. So does this imply homology or not, right? So this is a question that comes up a lot, especially when you're looking at organisms from completely different families, or usually families of higher order, okay? So this and lower levels of similarity might be indicative of homology, provided that either one or more of the following applies, right? So the first, the similarity extends over a long stretch of sequence and is statistically significant by criteria known to be reliable, especially by BLAST algorithm. So if it fits the parameters set by BLAST, then we could consider that that the genes or the proteins are similar, right? And the second, although the sequence similarity is low, if you still see the same pattern of identical and similar amino acid residues in multiple sequences, multiple means you're not looking at 5, you're looking at maybe 10 or 20, a lot more sequence, and you still see the same pattern or the identical pattern, then you can also say that genes or the proteins are similar. And lastly, the pattern of sequence similarity reflects the similarity between experimentally determined structures of the respective proteins, or at least correspond to the known key elements of one such structure. So especially in proteins, if you see the domains, the sequence in the domain area is similar, you can be quite sure that the proteins are going to function the same. So even if it's a very low similarity level, like let's say 18% or 20% similarity, but the similarity is you see the similarity mostly in the domains, then you can be sure that the proteins most likely are going to function the same way, right? So I've shown an example here, the second type, where although the sequence similarity is low, you see the same pattern of identical and similar amino acid residues, which is seen in multiple sequences. So these are actually ribosomal protein L36 sequence from a number of organisms. So we have bacteria, we also have yeast, rice, fruit fly, mouse, and human. And you can see all those in yellow have like about 90% of them actually have the same amino acid, right? And the one in black is common in all, right? So you see a similar trend in these, although very low similarity, but there's a similar trend. So we can actually say that this gene, ribosomal protein L36, are homologous, right? They are actually homologs in all the different organisms, right? So this is the question for you. Once we are confident that a particular similarity, either in gene sequence or protein sequence, is not spurious, that means it's not by chance, right? Is common ancestry the only explanation? So this is a question for you. Any answers? Are you all around?
Miss, can you explain again?
So the question is this: once you're confident that what you see, the similarity you see, is not by chance, or not by chance, is common ancestry the only explanation? That means, is it because they are from common ancestors, they have similar function, or can anything else be a reason for similar function in a structure or a gene or a protein?
Uh, Firstly, maybe because of the common ancestor, or secondly, I can suggest it's because of any genetically modified or genetic engineering done to the specific organism.
Okay, right. Let me see a chat there. Mutation. Okay, right. So yeah, remember I explained about convergent evolution and divergent evolution. So actually, common ancestry means divergent, all right? So actually the answer is no. Even when you're having convergent evolution, you can have completely unrelated organisms with genes and proteins, which can adapt and get the same function over the years, right? So convergence from unrelated sequences can also cause homology. It's not only due to common ancestor. So in the broadest sense, convergence is also observed between all proteins that contain disulfide bonds, which stabilizes their structure, or even between all enzymes that have the same catalytic residues. So even more prominent motifs associated with catalytic residues are found within different structural contexts and in all likelihood have evolved convergently. That means they don't share a common ancestor, but they have had similar environmental stress or challenge, which leads to mutations and evolution, right? Changes in your genes, which eventually leads to a similar function, right? Or similarity between these two organisms, right? Now, the next question, also for you, does structural similarity always reflect homology? You understand the question? That means when the structure looks the same, does it always mean it's homologous?
No, again.
No? Anyone says yes? See a lot of students. Lillian, what's your answer?
I think maybe
Maybe yes or maybe no?
yes.
Okay.
I'm not sure also. Sorry, doctor.
Okay, Fazla?
Maybe no.
No, okay. Tan Jingyi?
Maybe no.
No? Right. Let's try with some bioinformatics students. Hominia?
Maybe no, miss.
No. Priscilla?
I think no.
No also? Okay, so let's see the answer. So you understand the question, right? So every time you see structure similarity, that means the phenotype looks the same. Does it always reflect homology? Okay, so the answer is yes, especially when it comes to proteins. Usually, if you have a similar structure that spans at least one complete domain, one complete functional domain, most likely does. Okay, so this is what actually we use to do structure comparison, to study proteins using structure comparison methods. So they have softwares for it. They have WAST and DALI. So they actually use structure comparisons to study proteins function. Okay, so thus the general rule is of structure homology correspondence seems to be straightforward. Proteins that have the same fold will have actually the same structure, and therefore they have the same function, right? And they are known as homologs. Okay, so remember this: protein domains that have the same fold according to structure classification systems will mean that they have the same function. Okay, and so they are known as homologs. Right. So that's all for today's class. Actually, it's a short class. So do you all have any questions? Do you have any questions?
No, doctor.
Okay, so I hope you all can go through the practical video that Dr. Siva has given, and also the tutorial, the lectures he has given in the next 2 hours. So I finished quite early. You can go through them. And if you have any problems in getting into your Linux VirtualBox and trying it out, please let us know. Okay. So I will see you the week after next. So next week is your mid-sem break. So to those who are celebrating Deepavali, happy Deepavali. And for the rest, have a nice semester break. And I will see you the following week, the week after next. Right?
Happy Deepavali, Miss.
Happy Deepavali, doctor. Thank you.
Happy Deepavali, doctor. Thank you.
Thank you, doctor.